Bismillahirrahmanirrahim. My name is Fatma from DS Biotechnology, and today my topic is of presentation of proteins. Proteins are polyamides, and their monomeric units are composed of about 20 different alpha amino acids. Basically, when we break proteins, we get alpha amino acids. The exact sequence of different alpha amino acids along the protein chain is called the primary structure of protein. A protein primary structure, as its name suggests, is of fundamental importance for the protein to carry out its particular function, growth, development, etc. The primary structure must be correct. We shall see later when the primary structure is correct, the protein polyamide chain folds in particular ways to give its shape, its need for its particular task. Means in proteins that its primary structure is very important for the specific functions at specific level in DNA, in RNA. Folding of the polyamide chain gives rise to a higher level of complexity called the secondary and tertiary structures of the protein. Quaternary structure results when a protein contains an aggregate of more than one polyamide chain, means they have a complex structure of primary, secondary, tertiary. Hydrolyzed proteins with acid or base yield a mixture of amino acids. Means simple hydrolysis of proteins give us a mixture of different alpha amino acids. Here, this is a structure of an amino acids, and this is a proportion of a protein molecule in amide peptide linkage between the amino group. This is the first primary structure of protein. Here we can see sequence of a chain of amino acids. These are the amino acids that form a long chain and called as primary structure of protein. This is a beta sheets and this is called alpha helix, and they make a secondary structure of protein, and when they combine they form this type of a structure called as tertiary, and quaternary structure contain one or more than chain of amino acids. Extraction of proteins. Proteins are extracted in different methods from plants and animals and also from microbes. That when hydrolysis yields amino acids. So amino acids are the important in protein to function properly. Somehow the extraction and purification of protein is complex. It means a single replacement of amino acids can destroy its function. Hydrolysis of proteins yield different alpha amino acids. Although hydrolysis of natural looking proteins may yield as many 22 different amino acids, the amino acids have an important structural feature with the exception of glycine. Those molecules are chiral. Almost all naturally occurring amino acids have the L configuration at a carbon. That is, they have the same relative configuration as L glyceraldehyde. Sorry. Hydrolyzed protein yields different alpha amino acids. This is an amino acid, usually an F alpha amino acid. This is an L glyceraldehyde, S glyceraldehyde, and this is the Fischer projection of an L alpha amino acid and L glyceraldehyde. Fischer projection means the presentation of two-dimensional structure of a molecule. The presentation of two-dimensional structure of the molecule which have three-dimensional molecule structure. Synthesis of amino acids. Two amino acids are synthesized after the polyamide chain is intact. Hydroxyproline, present mainly in collagen, is synthesized by oxidation of proline, and cysteine, present most in proteins, is synthesized from cysteine. These are the amino acids. The conversion of cysteine to cysteine requires additional comment. The SH group of the cysteine makes a cysteine a thiol. One property of thiol is that they can convert it into disulfide by mild oxidation agents, and is that reversible, can be reversed by mild reducing agents. This the thiol is converted in disulfide. Here the cysteine is converted into cysteine, and disulfide linkage can be uh, present due to the oxidation, and it is reversible when some reducing agents attacked on it. Amino acids at dipolar line. Dipolars means they have negative and both positive charges. Amino acids contain both a basic group NH2 and acidic group CO2H. In the dry solid state, amino acids exist as dipolar ions to form a form in which a carboxyl group is present as a carboxylate ion, and CO2 and the amino group is present as an ammonium ion, NH3. Dipolar ions are also called as deuterions. In aqueous solution, an equilibrium exists between dipolar ion and anionic and cationic form of an amino acid. Here we can see in aqueous solution, this is a cationic form, this is an ionic form, this is negative charge. Here in ammonia we can see a positive charge, and this is a dipolar ion where oxygen have negative charge and ammonia ion have positive charge. The Strecker synthesis of amino acids. Treating an aldehyde with ammonia and hydrogen cyanide. Here aldehyde with ammonia and hydrogen cyanide produce an alpha amino nitrile. Here alpha amino nitrile. When this will hydrolysis of nitrile group of an alpha amino nitrile convert later to an alpha amino acid. This synthesis is called the Strecker synthesis. Here the alpha amino nitrile is converted into alpha amino acids with the when we heat it in the presence of water. Hydration of proteins. Ninhydrin is the hydrate of indane 1,2,3 trione with extraction of proline and hydroxyproline of all the alpha amino acids found in proteins react with the ninhydrin to give the same intensely colored purple anion. We shall not discuss here the mechanism, but notice only what portion of the anion that is derived from a alpha amino acid is the nitrogen. Here we can see the indane 1,2,3 trione is converted into ninhydrin, which is a same intense colored as purple anion. Reaction of the protein. The Maillard reaction of the amino group of lysine prevails in the presence of reducing sugar. This is an, for example, lactose and glucose, which yield protein bound n-deoxylactosyl 1 lysine and n-deoxyfructosyl 1 lysine. Lysine is not biologically available in these form, means in these form it is not available biologically. Acidic hydrolysis of such primary reaction yield lysine. Here we can see acidic hydrolysis in the presence of NaCl at 100 degrees Celsius for 24 hours. It yields lysine, furanocin, and pyridosine, as well as the degradation product of furanocin and pyridosine in a constant ratio. Isomerization of isoleucine. The reaction with L-isoleucine is of analytical interest. L-isoleucine is isomerized to D-alloisoleucine. Here, L-isoleucine is isomerized into D-alloisoleucine. Here we can see the chloride ion, and so has a retention time different from L-isoleucine, making its determination possible directly from an alpha from an amino acid chromatogram. Here, the continuous isomerization of D-alloisoleucine make it isomerization of L-isoleucine make it D-alloisoleucine, which is a dice. Stereoisomers mean it is non-mirror image and don't have identical isomer. Heating of protein in dry state and neutral pH result in the formation of isopeptide bonds between the amino group of lysine residues and carboxylic group of asparagine and glutamine residues. Here we can see formation of peptide bonds in the presence of ammonia. Oxidation reaction, oxidative change in proportion primarily involve methionine which reacted form methionine sulfide. Here we can see the formation of sulfoxide. So methionine is converted into methionine sulfoxide by the oxidation reaction. Protein metabolism, first of all, phenylalanine, which is an amino acid, is converted into tyrosine by the enzyme phenylalanine hydroxylase. Then these are the structures of these molecules. Then the tyrosine is converted into para-hydroxyphenyl pyruvic acid with the help of an enzyme called tyrosine transaminase. Here the alpha ketoglutarate is converted into glutamic acid. Then in step 3, the para-hydroxyphenyl pyruvic acid is converted into dihydroxyphenyl acetic acid, which is called as homogenetic acid with the help of an enzyme called para-hydroxyphenyl pyruvate hydroxylase. Here the oxygen is converted into carbon dioxide and it is known as ascorbic acid. Here this is the cleavage site. Then the dihydroxyphenyl acetic acid means homogenetic acid is converted into mallyl acetic acid with the help of an enzyme phenylalanine hydroxylase. With the help of enzyme homogenetic acid oxidase. Then it will convert in the mallyl acetoacetic acid into fumaryl acetoacid with the help of fumaryl acetoacetate hydrolase enzyme. Here, in the presence of water, it will yield two molecules, acetoacetic acid, which will go to the ketogenic pathway for the energy production, and the fumaric acid, which will go into the glucogenic pathway for the energy production. And in this way, the protein is metabolized into two molecules for further energy production. Applications of proteins. Therapeutic proteins provide an important therapies of variety disease such as diabetes, cancer, infectious diseases, hemophilia, anemia. Common therapeutic proteins include antibodies, Fc fusion proteins, hormones, interleukins, enzymes, and anticoagulants. They play a critical role as components of blood, muscles, enzyme, hormones, and hormone receptors, in addition to other biological roles in sustaining the function of living organisms. Protein also play an important role in food, providing texture, taste, flavor, which are essential for food selection. Proteins are considered useful for imparting gloss on a metal surface, softness, and manageability because of their substantivity. And hydrolyzation of proteins were proposed of permanent weaving treatment to prevent damage to hair fibers because of their amphoteric and buffering properties like keratin treatment. Here the biological function of proteins. There are many different types of proteins in our bodies. They all are serve important roles in growth, development, and functioning. The examples like enzymes which are involved in our metabolic pathways in our digestion that facilitate our proteins that facilitate biochemical reactions. For example, pepsin is a digestive enzyme in your stomach that helps to break down proteins in food. Means they break the peptide linkage in amino acids. Antibodies are proteins produced by the immune system to help remove foreign substance and fight infection. DNA-associated proteins regulate chromosome structure during cell division and play an in important regulation gene expression. For example, histone and collagen proteins. Contractile proteins are involved in muscle contraction and movement. For example, actin and myosin, which are present in muscles for the movement, contraction, and relaxation. Structural proteins that provide support in our body, for example, proteins in our connective tissue like collagen and elastin. Hormone proteins coordinate bodily function, for example, insulin controls our blood sugar concentration by regulating the uptake of glucose into the cells. Insulin is a protein. Transport proteins move molecules around our body, for example, hemoglobin that transport oxygen throughout the body. Thank you.